Okay, very good evening, my dear friends. Am I audible? Yugandhar sir, am I audible? Yugandhar sir, very good afternoon. Today, our resource person, Associate Professor, Dr. THIRUNAHARI Yugandhar sir, working in Kakatiya Government College, Autonomous, Anamakonda, Warangal. Sir is going to explain about the most important topic, not only in our botany, but also it is very helpful in biotechnology, genetic engineering, and also the disease detection of the disease and the control of the disease mechanism, and also the vectors, the plasmids, different types of the plasmids, the structure of the plasmids, and the artificial plasmids or synthetic plasmids, and the significance of the plasmids in biotechnology. These are the most important topics, synoptic points related to today's class. So in this regard, we would like to invite Dr. THIRUNAHARI Yugandhar sir. Please welcome sir and start your session. Yugandhar sir, please start.
Okay.
Start చేయండి sir.
Sir. Hello. Now starting. Now ready. So today, the discussion with the plasmids and their types. What are the role of the plasmids in biotechnology or gene cloning? So what are the plasmid is a additional genetical material of bacteria, virus, yeast, and also prepared from the artificially. These plasmid is major important role in gene cloning. The gene cloning is depends on the two types: natural gene cloning or artificial. The nature means vector-mediated gene cloning. The vector is called a plasmid, cosmid, phagomids, bacteriophage, bacteriophage. So several number of the vectors. Out of these all vectors, the plasmid is important for gene cloning. So this plasmid is additional genetical material of bacteria, which are first reported by the Heidelberg scientist. In addition to the bacteria chromosome nucleide. So this also got a bacterial DNA, but here is a plasmid. Plasmid is a circular double helical model, which is a functionally in the bacterial cell, not functionless, but which can add to the gene part, also called a foreign DNA, that can multiply in artificially, naturally. In addition to the bacteria chromosome, bacterial cells are normally containing genetic elements in their cytoplasm. These genetic elements are exit and replicate separately. Genetic elements, not complete DNA of bacteria. Genetic exits replicate separately from the chromosome and are called the plasmids. Separated, but nucleates
কোনো problem নেই।
and additional genetic material of plasmid both are
আশা করছি হবে, আমার তো মনে হয় হয়ে যাবে ওটা।
the very existence of plasmids in bacterial cytoplasm was revealed by Lederberg in 1952. He was Lederberg while working on the conjugation process in bacteria. So this conjugation is which major important role in bacterial reproduction, also importance to the new aspect for reporting the plasmid by Lederberg coined the term plasmid to refer the transmissible genetic elements that were transferred from one bacterial cell to the another that determine the male sex in bacteria. Pili. The pili is a special or conjugative process which can transfer the one bacterial DNA through the pili to another bacteria. That is called a place bacteria to minus bacteria. The place bacteria which are also called a male, also female is also minus bacteria that reported by the Lederberg scientist. So this one, the plasmid vector has majorly the 5 type of the functions: cloning plasmid, expression plasmid, reporter plasmid, viral plasmid, gene knockdown plasmids. It is also major. There are 5 types. What are the importance of these 5? We start that. Types of the plasmid: cloning plasmids. Purpose: used to clone. Clone means insert and multiply. Insertion means restriction endonuclease enzymes. When we add it to the plasmids, it can directly cutting and changing out DNA molecule into the vector. That is also Smith and Nathan was first reported the restriction endonuclease enzymes from Haemophilia influenzae, Escherichia coli bacteria. These enzymes are major role for directly attached to the empty part of E. coli part of the vector without cutting by man. Additionally, actually that enzymes are cutted and chain. So this DNA fragments features simple structure, contains an origin of replican parts. ORI part is located on the plasmid. That part is filled by restriction endonuclease enzymes. Then that the inserted DNA is when we transfer to the plasmid, that plasmid is cut and chain by restriction endonuclease enzymes and antibiotic resistance gene and multiple cloning site is also there. Not only single foreign DNA, it can add to the antibacteria. Also molecular markers, selectable markers, gene markers. The three types are added to inserted into the origin of replican part. That origin of replican part of bacteria which can transfer to the other bacterial cell, it can multiply under the culture. That bacteria is multiplied in several number. Every bacteria which can have a foreign DNA is multiplied part. That is the use ideal of a amplifying DNA sequence in bacteria. Ideal. The second one is also expression plasmid. The purpose used to express proteins of or RNA in host cells. Gene expression parts. Feature means the promoter sequence for initiating transcription. The transcription terminator to end a transcription also got a gene of interest. Often include the enhance the boost expression. It can further the use common in producing the recombinant protein or studying gene function. Recombinant protein or gene, what are the function to study this expression? That is called expression of the plasmid. This is also second type of the plasmid when uses. Third one, gene knockdown plasmids. This type of the plasmid purpose used to reduce or silence the expression of target gene. Silence means when we activated that, example, some growth of the bacteria. The growth of the bacteria, it can passes the fastly. That gene is represented or identified and silenced. by that gene is also gene knock down plasmids. Features often containing a short hairpin RNA or siRNA sequence target the mRNA of the gene to degrade it for block its translation. Use studying a gene function by observing the effect of the reduced gene expression. Observing and also reduce. This is also reporter plasmids. The third one, purpose means used to study the activity of the specific genetic elements. Example, promoter, also called inheritance. Features contains a reporter gene like a GFP, green fluorescent protein, special type of the protein, or luciferase. It can allow the visualization or measurements of the gene activity. Measurements means the size of the gene activity. The gene size and activity denoted this type of the plasmid useful for regulatory element in analysis. Gene regulation. First one is the gene expression. First one is also gene cloning, gene expression, gene regulation. The three type of the plasmids are reported. Then the viral plasmids. So it is purpose used for the delivery of genetic material into cells using viral mechanism. Viral mechanism means it is also the bacteriophage virus. So that the feature can be used to produce viral particles such as lentivirus, retrovirus, adenovirus, or AAV virus means adeno-associated virus. The four type of the virus which have called the plasmids activity, that's viral plasmids. High efficiency gene delivery to the target cells. What is the use gene therapy? Gene expression, first one, gene cloning. Second one, gene expression. Third one, gene regulating. Fourth one, gene therapy. Gene therapy means it is also treatment of the genes. Long-term gene expression studies and in a difficult to do transfect of the cells. So this is also fifth type of the elements. Then what are the physical nature of the plasmids? What is the physical? The structure of the plasmid is, which is also mapped there. Here map how many number of genes. What are their each function? Then the physical nature of plasmid is quite simple. They are a small double-stranded DNA molecules. Majority of plasmids are circular, but many linear plasmids are also known. But the linear is sometimes, but several plasmids having plasmids is a same circular manner. Naturally occurs plasmids vary in size. Naturally and artificially, two types. Based on the natural, few biotechnologists or biochemists which are prepared the artificial plasmids. Example, PBR322, plasmid, Bolivar and Radicus. These two scientists were prepared in artificially. Nature plasmid is based to prepare artificially based on the size of the kilobase measurements of the size of plasmid. Then naturally occurs plasmid is a vary in size, approximately 1 kilobase to more than 1 megabase. And typically plasmid DNA is considered to the less than 5% in the size of bacteria chromosomes. Small, very small, and it is a total 1 kilobases. Now, artificially 5 kilobases, 10 kilobases are plasmid is prepared by the biochemistry scientist. Most of the plasmid DNA isolated from the bacterial cells exist in the supercoiled configurations, which is the most compared from the DNA to exist within the cell. The copy number refers to the fact different plasmids occur in different cell number. The plasmid number is not constant in each bacterial cell. Up to 100 plasmids are there, single bacterial cell. That plasmids are all which are freely on the cytoplasm of bacteria. Then only center part of the bacteria is a nucleide. That the nucleus is also called single nucleus or single DNA parts. That is called linear part. Additionally, is also circular. Some plasmids are present in the cell in only 1 to 3 copies. Whether other may be present in over 100 copies. 100 copies of the plasmids are present in the highest number. Minimum 1 copy of plasmid is highest is also 100. The copy number is controlled by genes on the plasmid and by interaction between the host and plasmids, host cell and plasmid cell base. It can number is also different. So what are the properties of plasmids? What are the characters? So they are specific to one or few particular bacteria, specific. Then they replicate independently of the bacterial chromosomes. Bacterial chromosomes and replication of the plasmid is also different. For example, bacterial binary fission, also called reproduction of the asexual and sexual reproduction. The plasmid is a separate divides and nucleus are separate divides, not only combined. Means two genital materials are present in the bacteria. But in man, which have three type of genital materials, nucleus genome, mitochondrial genome, also called as cytoplasmic genomes are present in man. But here in the bacteria, only two genomes like nuclear genome of a center part of nucleide, then peripheral is also plasmid genome, also called a plasmid DNA. Plasmid DNA. So this is also the two type of the measure. So they code for their transfer. So they act as episomes and reversibly integrating into the bacterial chromosomes. So they may pick up the cancer, certain the